Hi, and welcome to the Power Hungry Podcast. I'm Robert Bryce. On this podcast, we talk about energy, power, innovation, and politics. And we're going to talk a lot about politics and innovation today with my guest, a return guest on the Power Hungry Podcast, Matt Ridley. Matthew, welcome back to the Power Hungry Podcast.
Robert, it's great to see you, and Happy New Year to you.
Thanks very much. So you've been on the show before, but you know I have guests introduce themselves. I need to tease it. You're the co-author. The reason why you're on today, the co-author with Alina Chan of the book Viral, The Search for the Origin of COVID-19. But if you don't mind, introduce yourself to the audience.
Yeah, well, I'm a science writer. I was a biologist many years ago. I then became a journalist and ended up writing books about evolution, genetics, molecular biology, as well as economics and environmental subjects. And when this topic came along, this pandemic, I got more and more intrigued by its origins. I discovered that the story was getting more confused, not less. And whereas it looked like it was going to be a simple question like SARS of finding a connection with a market, that then became more and more obscure and difficult to pin down. And eventually I decided to approach a brilliant young scientist by the name of Alina Chan at Harvard and MIT, who was uncovering interesting facts about the virus and its origins, and suggest we join forces to write a book. Because although I know a lot of molecular biology, I don't consider myself up at the latest frontiers of the topic. And she's working in vector genomics at Harvard and MIT, so you don't get much better than that. And
this is your 9th book, if I count correctly, is that right?
No, you don't count correctly. It's my 10th book.
Well, I'm still learning that. So tell me, what number is this?
It's my 10th book.
Your 10th book, thank you.
The very, very first book was an account of US presidential politics by a journalist on the campaign trail, i.e. me, in 1989, which luckily nobody reads now.
Well, so let me thank you. So this is your 10th book. So it came out November 16th, and there was a lot to talk about. But what has developed since you finished the manuscript? What are the Omicron being the obvious one, but what is this has been such a fast moving story all along, and you've captured a lot of the story from December and late 2019 up to mid 2021, where you had to submit the manuscript, or late fall of 2021. What's happened since since you stopped writing and since the book came out?
Well, we drove our publishers mad by demanding the opportunity to make additions and corrections right up to the middle of September, just 2 months before publication, which was an unusual thing to be able to do. But still, of course, as you say, we missed stuff. And one of the biggest stories that broke just after we'd finished adjusting the book was the discovery of a virus in Laos, in the country of Laos, that is slightly more closely related to SARS-CoV-2, the virus that causes the pandemic, than the previous most closely related one. It's 96.8% instead of 96.2%, basically, at the genetic level. And that's very interesting because it seems to exonerate the one that the previous one, which had been found in the freezer of the Wuhan Institute of Virology.
But this is another bat virus.
This is another bat virus. It was found in a bat in a cave in Laos. But the interesting feature of that story is that Lena and I and various other people immediately pointed out that the scientists who'd been searching for SARS-like bat viruses had been working in Laos as well as China, and had been sending those viruses to the Wuhan Institute of Virology. The EcoHealth Alliance, which is the US foundation that works with these Chinese scientists, was very explicit about the fact that if it found viruses in Laos to analyze, it would like to send them to its partner in China to analyze, even though the Chinese weren't, it wasn't a Chinese project at all. And when I pointed this out in an article after the book came out, the EcoHealth Alliance spokesman on Twitter said, "Oh no, we never did that. We applied to do it, but we never did it." So I replied, well, okay, how do you explain the fact that there are quite a lot of entries in viral genetic databases showing viruses collected in Laos by the EcoHealth Alliance and deposited at the Wuhan Institute of Virology. Here's one. I gave an example from a database. Deathly silence ever since from their spokesman. And I've prodded him about once every 2 weeks since then, saying, look, please, could I have an answer? Because you're claiming that this couldn't have happened. Here's evidence it did happen, which in the evidence is important because it says if the closest relative was in Laos, not in China, there's still a connection to Wuhan.
Right.
It's still the one city on planet Earth to which viruses were being taken from bat caves and deposited in sample collections.
And where some of the earliest human infections were detected was in Wuhan.
And it's, of course, where the pandemic began. Exactly.
Well, so, I mean, what's remarkable about your book, I mean, and I got to tell you, look, it's so complicated. I mean, you're talking about so many different strains and so many different genomes and the different parts of it. I mean, it's really a detective story with a cast of characters that is quite large. But I want to come back to that because you mentioned Alina Chan, and I don't know, I've very seldom co-authored things, but I mean, for you to approach her, I mean, how did that work? I mean, I wanted to start there because I just a remarkable collaboration where, in fact, you were here in Austin in late October, and you said you had never met her, and you met her in mid-November for the first time face-to-face, right? I mean, it's a very unusual collaboration.
A week after I saw you, yeah.
Right.
And, it was very moving for both of us. You know, we met in an outdoor cafe and in Boston, in Cambridge, and I saw her across the street and approached her and waved, and and we unpacked a pack of the books which had just reached us. We hadn't seen the books at that point. And so, that's a remarkable thing to do, to write a book with someone. You never meet them. And then you do meet up when the book is finished. How did it work? Well, as I say, I approached her. I was thrilled and delighted that she said yes. I'd already had enough correspondence with her to know that she was an extraordinarily intelligent, thoughtful person. And she had taken to Twitter, to social media in particular, to start a conversation with other scientists and other, amateur detectives about what we could learn about the origin of this virus. And she thought it was a very important topic. And on Twitter, she rapidly developed a reputation as the most thoughtful, but also the most careful person speculating about this topic. And she'd had to interrupt her own career, her own experiments, because the lockdown prevented her going into the lab, and that's what got her started on this story. The head of her lab, Ben Deverman, was very sympathetic and thought she was doing a great job. And so she's now, testifying before the House of Commons in London, before the Congress. She's the kind of leader in this world, but she's 33 and I'm 63. So an old British science writer teamed up with a young American scientist to produce a book. You could say I'm the writer, she's the scientist, so the facts are hers, the prose is mine, but it's not quite that simple. She writes pretty well too, and I have my opinion about various facts and what they mean and how they should be interpreted. But I wrote the first draft, she completely rewrote it. I then started rewriting her rewrite and so on. We did that several times. And then we did a lot of updating and so on. And actually, it worked really well. The time difference was helpful, interestingly, because I would, finish a rewrite of a chunk of text at evening time mine and dump it across the Atlantic for her to have a go at in her daytime.
Well, I'm just curious about how is you working in a Word document with track changes? I mean, I'm just curious about that mechanics of that. Was that you're just exchanging, or you working on Google Docs, or what was the format there? Because the intricacy of all the data was the part that I thought, man, how did they keep it all straight? Yeah,
well, the working document was the book itself, obviously. The various chapters and so on. And it was we had to have immense security to make sure that no one else was eavesdropping on what we were doing.
Right.
But we did use at times we were sending Word documents. At other times we were working on a joint Google document.
I see.
I'm trying to remember, actually. It's a good question. But we were communicating by a secure email and by secure phone calls. We really didn't want anybody listening in or reading anything. And almost nobody saw the manuscript until it was published. We didn't share it with, normally when you write a book, as you send it out to people for pre-review and comments and blurb quotes for the cover and things. We didn't do any of that. We wanted to keep our powder dry until the world saw it.
So, well, there are a lot of things I want to talk about, but since you mentioned effectively meeting or being introduced to Alina Chan on Twitter, one of the things that was remarkable about the book and your reporting on it was the role that individuals and non-scientists had in helping crack the whole story. You cite Charles Small, who I think is a Brit or an American, I can't recall, but he's a business consultant who advises corporations on different, travel and different conditions in other countries. And then the remarkable story about Francisco de Ribera, who's a Spaniard who lost his job and then delved into he was a database whiz who was able to publish then on Twitter the genomic tree of the viruses, which was incredibly important. But he was just I'm not saying a regular guy, but he wasn't with some fancy university or didn't have a big background in this. I guess the question I had is this the question that I wrote down after I wrote all this is this the part of the way that science is now being done in an open source environment?
Well, it should be. It's very important that we all use open source to crowdsource solutions to problems, to draw on expertise wherever you can find it. And there is a long and noble tradition of citizen science, of people gathering data, just amateurs gathering data. For example, in this country, and I think in the US too, bird watchers have been enlisted into, systematically recording data so you can detect population trends and so on. So citizen science is not new, but the internet enables it to happen in new ways. And if you take Francisco, it's a really good example. He was a fantastic source for us in our book, very, very helpful to us. As you say, he's an IT consultant, a tech consultant really in Madrid, and he lost his job. He got interested in this stuff. He had the experience that when you audit a company's accounts, one of the things you look for is missing serial numbers of invoices, right? So if there's a series of invoices and there's two missing, you ask to see those two missing invoices, and they turn out to be the ones that show corruption or whatever. Well, he said, I want to find the serial numbers of every virus they found, because they were actually quite systematic at the Wuhan Institute of Virology at naming viruses with a number when they found a new virus in a bat sample from a cave in Yunnan, usually Yunnan. And they started a new series of numbers with a different beginning number for each collection trip. So they'd go to the Shitu cave and they'd collect 35 types of viruses or something, and they'd start them with the number 7. And then they'd go to the Mojiang cave and they'd start them with the number 4 or whatever it might be. And they would then deposit some of the details in genetic databases. They would mention others in published papers or in grant proposals. And from all that information, Francisco was able to work out which viruses came from which caves, which order they were collected in, which ones were missing, quite importantly.
And it's important that they had changed some of the nomenclature on how they were tracking the viruses as well, which he also figured out, which was another remarkable part of the puzzle. No?
Yeah, well, one of the one virus whose name they changed sometime between 2019 and early 2020, because we know they were still referring to it by the old name in 2019, but by the February of 2020 they're calling it by a completely different name, is the one now known as RETG13. Which was the closest relative to SARS-CoV-2. Now they say, oh, well, we changed the convention of how we name these things. So of course it got renamed. Well, what a coincidence that you rename the one virus that is closely related to this pandemic and whose origins you obscure in the paper where you publish it. You say, We found it in Yunnan. You don't say when, you don't say where, you don't say that you've published anything about it before, you don't say that it was associated with the death of 3 people and the illness of 3 others in an outbreak of viral pneumonia that scared the bejesus out of scientists at the time in 2012, sorry. These were important details, and it took people, these sleuths like Francisco, the Seeker, a guy in India, a guy named Baba L'Elephant in France, Charles Small, as you say, he's the guy who worked out that it was on the 12th of September 2019 that the Wuhan Institute of Virology's virus database went offline.
Right.
2 a.m. Now, that might not be a significant moment. That might not be, it doesn't sound like when the pandemic could have started. It sounds much too early to me. But it may indicate that there was something going on, like a rehousing of the samples. They might have been moving them from the old site of the Wuhan Institute of Virology to the new one, which was happening around that time. Something happened around then that could have led to a chain of events that could have led to a leak, to an accident. If this was a Western lab, we'd be all over it. We'd be asking the scientists, look, sorry, we want your logbooks. We want to see exactly what happened. That's the best way for you to prove your innocence, by the way. If this didn't come from an accident in your lab, then you've got every reason to share the database. And that database has stayed offline. It's got hundreds of thousands of entries of, sorry, no, I've suddenly forgotten the number, but it's got a very large number of viruses in it. Any one of them could be relevant to this pandemic. We keep being told they're not. Well, then show it to us. Why don't you? And the point of accumulating all these viruses was to be ready for the next pandemic. Well, which pandemic are they waiting for?
Right. Well, so I want to back up, because that was one of the things in the book that I thought was, a lot of intriguing things, but just that the crowdsourcing, the citizen science, as you say, ended up being a crucial part of understanding the origins of the virus and forcing, in many cases, the Chinese to be more transparent. Now, they haven't been transparent, right? They've been minimally transparent, and the transparency has been, in many cases, forced upon them when confronted, and the same with EcoHealth Alliance. But let me come back to those issues. But I wanted to start with, because you really start the book at Mojiang, the Mojiang mine. Why is that mine so important? You mentioned it earlier in 2012. That was where a lot of these early viruses were discovered. Why is that mine important in the story of COVID?
Well, the virus most closely related to SARS-CoV-2 was found in a freezer in the Wuhan Institute of Virology at the start of the pandemic. It had been collected from a mine shaft in Yunnan at a place called Mojiang. The reason the scientists from Wuhan were in Mojiang, which is 1,800 kilometers by road from Wuhan, it's not next door. It's a very, very, it's London, New York to Orlando. It's that kind of distance. And the reason they were there was because the year before, in 2012, some people had been shoveling bat guano out of a horizontal chamber of an old copper mine, probably to sell the bat guano, or possibly to sell the bat guano, possibly to reopen the copper mine as a copper mine. We're not sure. We don't know the details of that. And 6 of them had fallen very sick. And they'd been fallen so sick that they had not been treated in local hospitals. They'd been taken all the way to Kunming, 6 hours drive away, which is the capital of Yunnan. And there they'd been reported on by the most senior virologists in China, not just from the Wuhan Institute of Virology, but the guy who really sort of was the hero of the original SARS epidemic 10 years before. Because everyone was convinced that what had happened was they'd caught a bat virus. And if they had, it would be the first occasion ever when one of these SARS-like viruses had been caught directly from a bat. Because in the case of SARS, it looked like it had been caught by palm civets, which had then given it to people. So people weren't catching it mostly directly from bats. But here was a case where it looked like it had spilled over directly in a man-made cave and it wasn't the same virus. It wasn't SARS, but it looked like it might be closely related. So it's very interesting that they found a SARS-like virus in that cave, but it wasn't quite the same one. So you'd have thought, this is exactly what you're looking for. If you've got a whole research program about forecasting the next pandemic based on sampling viruses in wild bat populations to try and identify viruses that might threaten a future pandemic, this is exactly the kind of case you'd be very interested in. So it's unlikely that sample festered remained neglected in a freezer for all these years. It's more likely that they took it out and studied it. And we later found out, again thanks to Francisco Ribera, that they had found 8 other viruses 2 years later in the same mine shaft that were also very closely related to what we now call SARS-CoV-2. And they admitted that 6 months later, after Francisco had worked this out. So you've got the 9 most closely related viruses in the world to one that's causing a global pandemic in your lab, which you collected from a place where 6 people got sick. It's quite relevant, all that information. And you'd like to know what's been going on with these viruses in the lab ever since. Now, in the case of the first one, they implied in their paper that they had not touched it for 6 years, but had simply brought it out when the pandemic started, sequenced it, and said, "Oh, look, it's the same," or "It's very similar. It's 96% the same as the pandemic virus." In fact, we now know that they had deposited parts of its genome sequence in international databases in 2018 and 2017, right? So no, it hadn't been lying neglected in a freezer, as Peter Dazak kept saying it had. It had been studied over this time. Now, if you're studying it, if you're bringing it out and sequencing it, then you're handling it in the lab, and there is a risk that you're going to infect people in that process. Now, it's not SARS-CoV-2. Get this straight. This is 96% the same. It's not 99% the same, which is what we need to find. But if they're handling lots of viruses from that site, all of which are quite closely related to SARS-CoV-2, maybe there's another one in there. Maybe there's one they didn't detect. Maybe this one isn't actually a real virus. It's just a combination of 2 different parts of a virus. And if you put them together in a different way, you get one that is 99% the same, etc. So, maybe they recombine in the lab. So all of that is highly relevant to ruling out or ruling in the possibility that SARS-CoV-2 was in a lab and was being experimented on, maybe unwittingly, in the months leading up to the beginning of the pandemic.
Well, so I want to talk about Dazak as well, but throughout the book, you make this point over and over, you and your co-author, Alina Chan, that when there was a choice for transparency, the Chinese officials chose not transparency. They chose to obscure. So the question, why? Well, you've thought about this a lot, I'm sure, over the last year. Is this an embarrassment for China? Why have they been so? I think the word is dishonest about the whole event. What lies at the root of this? I mean, you can only conjecture here, but what's your guess?
Yeah, well, Alina and I are both a bit reluctant to do too much conjecture because that can get you into trouble, and we try and stick close to the facts. But I think in this case, what you need to take into account is that under a authoritarian regime like the Chinese one, people are told very clearly what they can and can't do. And if an edict comes down from on high, which it did right at the start of the pandemic, nobody can share any information about this new virus, this was in January 2020, on social media or published in the scientific literature or in other media without permission of senior party officials, right? You just can't. In that case, people are going to be cautious about what they say, about what they reveal. They're going to err on the side of caution, because if, say, you publish something saying, look, by the way, this is the same virus as we found back in we've called it something else, but we changed its name, and 6 people died, and it was in a mine shaft, and they were shoveling back guano. If you add all that into the paper, and your boss comes down the corridor and says the party's unhappy with you having done that, it's quite serious,
career ending, or
exactly.
maybe freedom ending event.
Yeah, and we include a little story in there, which is a little whimsical, about a leak that did happen in 2020. It's a leak of leopards. Okay, so a farmer in the middle of China in May 2020 is in his field, and suddenly he sees a leopard. Leopards don't live around here. And everyone says, you must have been drinking. And he says, no, I saw a leopard. And then another farmer sees a leopard. And then, and then it gets the local paper picks it up and says, people are seeing leopards in the middle of China. We don't have leopards around here. What's going on? So they call the local zoo and they say, have you lost any of your leopards? And the local zoo say, no, not a chance. Well, 3 weeks later, the local zoo has to come clean and admit, yes, actually, we did lose some leopards. They've gone missing. Now, if you can try
not like losing your iPhone or something, or my ballpoint pen. Oh, yes, there's some leopards suddenly not here.
Try to cover up the loss of leopards. Think how much easier it is to fall into the habit of not being transparent about the loss of a virus. And probably the unconscious loss. The other point you need to bear in mind, of course, here is that when SARS infected lab workers, which it did on at least 4 occasions, twice in Beijing, once in Taiwan, once in Singapore, on 3 of those occasions, we don't know how it happened. All we know is that someone working on SARS in a lab got infected. There wasn't a dropped test tube or a punctured protection suit or something like that.
One of the other, we've talked about the complexity of and your exchanges with Alina and your need for security and so on. We've talked a little bit about Francisco de Ribera and Charles Small. Who are the heroes of this story?
Well, we say at the end of the book that the people we think are the heroes are partly those internet sleuths, those guys who, with no official backing at all, just found out really important information about the origin of the virus. The citizen scientists, the open source analysts, I think is a good word to use for them. But also partly the Chinese workers who blew the whistle early on. The ophthalmologist who published something on social media saying, look, guys, in our hospital, there are cases of infectious viral pneumonia. It looks like SARS. It's very scary. Please take care and let us know if you know anything more. And for that, he was punished. He was given the most terrifying, when you read it, document to sign, saying that he was an evil and disgraceful person who should never be seen in society again. And he lost his career, and within a month he's dead of SARS. Of SARS-CoV-2, of COVID. So he and the other scientists in those hospitals in the early days who realized that they were dealing with something, they are also the heroes. And some Western scientists are heroic too. People like Richard Ebright, David Relman have consistently said, sorry, we can't rule out a lab leak. We need to know what's going on here. And they've come under immense pressure from their scientific colleagues to toe the line. And the line was, no, we can rule that out as a conspiracy theory. Remember, we're not saying in the book that that's definitely the answer. We're saying a wildlife market origin is still possible. A lab leak is still possible. We can't at the moment find definitive evidence to distinguish between them, but the circumstantial evidence leans strongly towards the lab leak.
Well, and I it was remarkable in the book how restrained you were, and you made those points over and over, but you still didn't say, no, this is definitely the case, because you still can't know. But there are other parts of this story that are truly remarkable, and one of them that struck me early on was you point out that when it was within a day or so, only a matter of hours after the full genome was published on, what was it, January 12th of 2020, where once that was published, Moderna had modeled or designed a vaccine, an mRNA vaccine within a day or so. I mean, it's truly stunning about the ability of the company with the technology to bring to bear what was potentially a vaccine within such a short amount of time. Am I the only one that was slack-jawed by that? I mean, just how rapid it was?
No, I agree. And Greg Zuckerman has written a very good book about the vaccine race, as it were, with Moderna and BioNTech and AstraZeneca and Oxford in the UK, the degree to which Moderna was an incredibly ambitious entrepreneurial company with a brand new biotech technology that it couldn't quite make work, couldn't quite make safe, wasn't quite sure what the applications were, was thinking about cancer, but was beginning to think infectious disease vaccines might be a better way to go. And through its ambitious CEO, Stefan Bancel, latches onto this new outbreak and says, this is our opportunity to try and prove that this technology works and could produce a quick vaccine, effective vaccine, and a safe vaccine. And boy, they were right. I mean, I had Moderna for my booster, for my third jab about a month ago, and it sent me to bed for a day with severe chills. I just couldn't get warm in bed. My first two vaccines had been fine, but for some reason I had a bad reaction to that one. And those are exactly the symptoms that they kept getting when they tried injecting messenger RNA molecules into people. They couldn't get the dose right. Either they wouldn't get enough of an immune response, or they would get these severe reaction. And you know, so it's still an experimental technology, and they're just trying to get it just right. But boy, are we lucky to live in an age when that technology has come along, because it enabled us to get vaccines within a year, which is remarkable.
Well, and the ability to scale, and the ability to scale the production as well is miraculous as well. I mean, just that you can go and not just you're not making a few thousand, you're making hundreds of millions of them.
Exactly. And you couldn't do that with the old technology of making vaccines, which is to grow whole viruses and attenuate them. That takes much longer time. But if you're just generating messenger RNA, synthesizing messenger RNA molecules, then in theory you can do it much quicker.
Is this? I was reading something else. Someone else talking about RNA and that technology, and the potential for that. How this is something that just occurs to me as you're talking about it. But how promising is that field of RNA in terms of therapeutics and vaccines, and how far along are we in that field?
I'm not sure. I mean, I think it could go either way. It's possible that this will prove to be a quick and dirty way of getting a vaccine that half works against a horrible new threat, but doesn't produce the sort of long-lasting immunity that you really need if you're going to defeat a chronic disease or something like that.
Which means we may have to keep getting boosters for years to come.
Exactly, unless we move to a different design of vaccine that gives you longer lasting immunity, et cetera. So it's possible this thing, it helps get antibodies, but it doesn't get the T cells going, or whatever, So it may be, in some senses, a semi-disappointing technology. But on the other hand, if you look at vaccine development before the pandemic, it was stagnating. It had moved nowhere since the 50s or 60s. Not quite true, but, there are regular complaints in the literature from 2019, as late as 2019, of people saying, why can't we speed up vaccine production? Why doesn't it get enough investment? Why is it the sort of Cinderella of the pharmaceutical industry? Isn't it shocking that it takes a decade to produce a vaccine against AIDS or malaria or whatever? Ebola, they got a couple of promising vaccine candidates, and then the epidemic petered out, so it wasn't possible to really test them, etc. So it's not easy to produce vaccines.
Well, you wrote, and we talked about this the last time you were on the podcast, in your book on innovation. You profiled the two women who pioneered. I'm forgetting what the vaccine for
Whooping cough.
Whooping cough. Yes, thank you. And that was in the 40s, and they worked at it for
30s. Yeah.
In the 30s, and they worked at it for a decade, and they were considered quacks, or more than a decade, wasn't it? Remind me of that.
Well, no, actually, they did it in 4 years.
Oh, okay.
But the point was that was pretty. I mean, that was fast for then, and it was fast for today, actually, and nobody believed them. Grace Eldering and suddenly forgotten the other one's name, but anyway, too much Christmas has fried my memory.
I couldn't remember what the vaccine was for, or the 40s or the 30s, but that's okay. But yeah, I mean, from designing a vaccine in a day compared to 4 years, I mean, that's still remarkably short time for, comparatively to what's happened in the last, 70, 80 years then.
Well, and of course, it just goes to show how incredibly portable genetic information is. That is to say, you don't need to send a sample from Wuhan to Cambridge, Massachusetts, where Moderna is based. You send a string of digital letters. You send a genome. You send 30,000 characters, and that a 10,000 word document, as it were, in 3-letter words. That's all you need to splash around the world for people to start working on the problem, because you can then, from that, you can synthesize a genome straight away. That's the extraordinary thing. We've made biology at that level into a digital science.
Well, it seems like you were perfectly prepared for this book. I mean, it seemed like all of the books I don't know about your career, but I just find one thing in my career leads to the next. It seems like you were in the perfect place to write this book, given your work on the genome and innovation and all these other things. It seemed like your expertise came in just in the perfect timing here.
You're right. That was one of the considerations that came into my mind, is that, I'd written a book called Genome, I'd written a book about nature and nurture, I'd written a biography of Francis Crick. I knew genomics. I was fascinated by it. I loved it. I wasn't as up to date on it as I could be, because it's so fast moving, but I felt that I could understand elements of this story. Also, there's a lot of natural history in here. Like you, I'm a bird watcher, but I'm also very interested in all forms of natural history. And so the whole subject of, the bats behind the pandemic, which was an article I wrote for the Wall Street Journal, was fascinating to me. It's not just any bats. These are horseshoe bats. There is one specific genus, Rhinolophus, of bats that carry these SARS-like viruses. No others, as far as we know. They don't live in the Americas, by the way. They only live in the old world. They're very diverse. There's about 100 species. They overlap enormously in species diversity in southern China in particular. You get up to 10 or even 20 species, sometimes sharing the same cave. They're very interesting little animals. They're the most, in some ways, the most sophisticated bats. They've got very high-end echolocation systems. They can navigate through dense vegetation at night on short, fast wings, using sonar, obviously. So, that the natural history side of it attracted me too. And, it is it's an important story, but it's also a very fascinating story. Why do viruses jump species? Why did this one arrive in the human species? When did it do so? We're obviously dry tinder for it, in the sense that it found our immense numbers and our tendency to travel a lot are very helpful to its spread. It was beautifully designed to cause a pandemic, because it could infect, it could cause very serious infections, but you could also transmit it pre-symptomatically for several days. So it was a fiendishly clever device, devised, well, mostly by Mother Nature. This is a natural virus. There's no question about it. It has the same set of genes as any other SARS-like viruses. There are basically 14 of them. It's not as if someone's designed it from scratch. But somewhere along the line, it's become adept at infecting human beings. And it did so pretty well before the pandemic started. That's the spooky thing about it. SARS wasn't like that. SARS had been infecting bats and then civet cats and other animals in markets, and it then started infecting human beings. And to start with, it's really not doing a very good job. It's not very infectious. It's not very easy to catch it from other people. And in the end, we were able to control it with non-pharmaceutical interventions. So although it was very dangerous, it wasn't particularly infectious. Whereas this one arrived in the human species saying, right, this is where I'm meant to be. I'm well adapted to this species. I'm better at fitting the ACE2 receptors on human cells than I am on bat cells, than I am on mouse cells. Where did it acquire that characteristic? Now, one possibility is that it's been in a lab where it's been infecting human airway epithelial cells, which are cells taken from human lungs that are used to grow viruses in the Wuhan Institute of Virology. Or it's been infecting humanized mice, which are mice with human ACE2 receptor genes in them, which have been used for in vivo experiments with SARS-like coronaviruses in the Wuhan Institute of Virology, among other places. So if there's, several generations of these mice have been infected with a virus, it's going to get good at unlocking human locks on cells.
Well, let's follow up on that, because you just put something, today's January 4th, and we're going to post this in this episode today, but I think it was yesterday, it was 2 days ago, you put on Twitter, a notice on a new paper that was talking about Omicron. There's evidence that it was mutated through mice, that there is the history, apparently, am I remembering this correctly, that Omicron had some history in generation in mice species. Am I remembering correctly?
Right. Yeah. Now, this is a new paper that's just been formally published. It was out as a preprint a couple of months ago, but it's now been peer-reviewed and published from a Chinese team, interestingly, analyzing Omicron, this new variant of SARS-CoV-2. And what they conclude is that it's descended from a relatively early variant of SARS-CoV-2, from a mid-2020 variant. It's not descended from a 2021 variant, if you see what I mean. Its common ancestor with the Delta variant, for example, is in the middle of 2020, not in 2021. So it's not a recent outgrowth. It's been somewhere for a year and a bit. Okay. Now, where's it been? Because in that time, it's acquired an enormous number of mutations, rather more than you'd expect. Most of them are non-synonymous. That is to say, they actually do change the protein as well as the gene. What the paper concludes is that the pattern of mutations that has appeared in that virus, in the Omicron variant, does not particularly represent the pattern you'd expect if it had spent that time in a human being, or a dog or a cat or a bat or whatever, but it does reflect the pattern you'd expect if it had spent time in a mouse. And this is because, these are the mutations you'd need to make to fit the receptors on cells in mice. So it looks like this virus was a reverse zoonosis. That is to say, we gave it to mice in 2020, and mice gave it back to us in 2021. That looks like what may have happened. Now, previously, we had thought, and Alina and I thought this was the most likely hypothesis, that it had been in immunocompromised human individuals, which there are many of in Southern Africa in particular, because of all the people who have HIV there and have not such well-functioning immune systems. And that might be where it had hung on and evolved. And that might still, that's not ruled out as a hypothesis. But this new hypothesis says that actually it might be mice. Now, it could be house mice. It could be that in Southern Africa, house mice have picked up the virus from us and then changed it and then given it back to us in 2021 in some pair of incidents separated by a year. Or it could be that these are laboratory mice that are, and remember, mice are used in laboratory experiments all the time. And it's not difficult to find experiments that have been going on in South African universities over the last year with mice and SARS-CoV-2. So We need to look into those.
Sorry to interrupt, but then there could have been potentially, not saying that, two different lab leaks. That one was with originally with SARS-CoV-2, and then it goes to Africa and then latches onto mice and then comes back out again. I mean, that there was. Am I misapprehending what you're saying here?
No, that's possible. Look, we need more information because, I don't want to be out there saying, oh, this might have been a second lab leak, and then along comes very definitive evidence that wasn't the case, and people say, oh, he reaches for a lab leak theory at the drop of a hat. That's not what I'm saying. I'm just saying let's look at all possibilities. Oh, and don't forget that there was a case of a lab leak in Taiwan a few weeks ago of SARS-CoV-2 from an experiment in a lab to a human being. Now, the reason that, in Taiwan, there isn't very much COVID, and so it's quite easy to detect these things, but a scientist working on the virus in a lab picked it up. That's not surprising. Lots of scientists are working on COVID in labs all over the world. They're bound to be picking it up. But it reminds us that even in this was a very high security lab. Even in high security labs, it's not easy to prevent this virus infecting people. I mentioned before, SARS infected people in labs at least 4 times.
So let's talk about the EcoHealth Alliance, because Peter Dazak makes many appearances in your book. I just looked again at the transcript of the interview he gave to Leslie Stahl in 60 Minutes in March of, I guess that episode was published in March of last year. And that was focused on the World Health Organization investigation in China. And Dazak says, well, effectively to Stahl about, well, we just have to trust the Chinese. But why is Dazak such a critical figure here? And has he been completely forthcoming?
Yeah, well, Peter Dazak is an entrepreneurial figure. He's a scientist, a parasitologist originally, but he followed his wife to the US in the 1990s and got a job running a project for a wildlife charity called the Wildlife Trust. And the job was essentially getting hold of money to look at viruses in wildlife and distributing that money to partners in countries where you might find these kinds of viruses, particularly in Asia, but also in Africa. And he built that program up so he became the main conduit between the American taxpayer-funded research budgets and the work going on with Chinese and other laboratories elsewhere in the world. And one of his biggest collaborators, one of his biggest recipients of the funding that he was distributing was the Wuhan Institute of Virology, and Xi Zhengli in particular. And she became a close friend of his. We've got lots of tweets of them talking about karaoke sessions, wishing each other well, and which is fine, nothing wrong with that. But clearly he is a major funder of, collaborator with, expedition leader with the scientists at the Wuhan Institute of Virology. That's a conflict of interest, right? And yet when he drafted, although he disguised his role in drafting, a letter to the Lancet in February 2020 saying we can rule out any possibility that this leaked from a laboratory as a conspiracy theory. When he did that, he also put out a statement saying, I declare no conflicts of interest. Now, the editor of The Lancet was asked about that in the House of Commons in London just the other day. And he said, well, we realized that statement was wrong immediately, but it took us 18 months to persuade Peter Dazak that we should change that statement. Well, I don't think it takes 18 months to persuade someone that if you're the editor.
Well, they don't need his permission they don't need his permission to make a correction.
Exactly. Now, has he been forthcoming? Well, I have tried to communicate with him numerous times. He has never once responded. He blocked me on Twitter. He has given interviews, as you say, but only under circumstances where he thinks the interview will be friendly. He obviously doesn't think I will be friendly anymore. but there are incredibly important questions one would like to put to him. And just to go back to this business of the 8 extra viruses that Francisco Ribera found. When he found that, he said to Peter Dazak, "I found these in the data attached to a paper that has your name on it. You're the senior author on," which was a summary of virus genomes, which was submitted before the pandemic, which is perhaps why it's out there. And it looks to me like these 8 viruses are very similar to SARS-CoV-2 and were probably found at Mojiang. Can you confirm that? And instead of replying to Francisco, he blocked him on Twitter. Now, this isn't the way scientists should be behaving in any circumstances, let alone in the middle of a pandemic, which is killing many millions of people. I'm sorry, but we need total transparency here. What were you doing with these viruses?
Well, in particular, if you're getting federal funding, then you should be making extra effort to be transparent, and instead it's been the opposite.
Well, that's right. For example, just as we were going to press with the book, a document dropped through a leak, which was a grant proposal from Peter Daszak, he was the name on the grant proposal, to DARPA at the Pentagon, asking for $14 million, which is a lot of money, to investigate the role of these viruses. And it included that they would do this in China with SARS-like viruses, and they would, if necessary, manipulate these viruses to put in a thing called a furin cleavage site, which makes the virus slightly more infectious and easier to grow in the laboratory. Now, that's very interesting because this virus has a furin cleavage site in it and none of its wild relatives do. So if we know that there was a team of researchers that was planning to or doing the insertion of a furin cleavage site into a similar virus, then we would like to know that. Peter Daszak's name had been on that document since February 2018. He knew that they were planning that experiment. He had never once volunteered that information until it emerged in September 2021. This is not good enough, for someone in receipt, as you say, of federal funding. Oh, and by the way, all the viruses collected under the EcoHealth Alliance projects ought to be available to the US taxpayer to inspect. If you've had US taxpayer funding to find these viruses, then why aren't they available? Well, a big chunk of them are in the Wuhan Institute of Virology database and are not available to US funding authorities on behalf of the US taxpayer. Again, that seems to us extraordinary.
Well, it's just, as you say all this and you're repeating it, and I read your book, but it's a scandal. Why hasn't he been forced to testify before Congress under oath? Why is he not in the dock being questioned by Congress? What is the next step here to get some accountability from this man?
Yeah, well, when Alina and I and Richard Horton appeared before the House of Commons Science and Technology Committee in December, they invited Peter Daszak to testify. He didn't. The US Congress could subpoena him. The Democrats who control the committees, on the whole, at the moment, are reluctant to do so. They still see this subject as a partisan one that Republicans are more interested in than Democrats. I think that's the only explanation I can think of. But it does feel to me that the time must surely come when information is, when testifying under oath is forced to happen by some of these people. And they should want to,
disclosure.
So you should, get out there and tell us why you think it didn't come out of a lab instead of letting us find this information drip by drip.
Or blocking on Twitter, or just making conscious efforts to obscure. But I guess the other part in reading the book and thinking about it, since you agreed to come on the podcast, I was thinking this has been the most consequential public health crisis in my lifetime. And you're 63, I'm 61. And yet we still are, as you mentioned, the partisan divide over this, and that it's been incredibly divisive. And in terms of, the vaxxers, the anti-vaxxers, the masks, the lockdowns, there's a very political divide between the conservatives and the liberals, to a left-right divide over trusting science and so on. It just seems like it is, amidst all the other divisions in society now, this has only compounded them. Well, and led to, there was a good piece in the Wall Street Journal today about this distrust of authority figures in science. It seems like this is only going to serve to make that distrust even greater because of the lack of common accepted facts. Does that rhyme with how you're seeing some of this? Because it just seems like it's made the divisions in society much worse.
Well, I think that in the current world, and I can't help but think it is got a lot to do with social media, everything gets polarized. I think that's been happening for most of the last decade. You know, for me, the moment when I first noticed it was the Scottish referendum in 2014, when I suddenly noticed that, you know, my Scottish friends were divided into people who hated each other, you know, in a way that I hadn't seen happen before in political arguments. You know, that it was very, very partisan. And then 2 years later, you've got Trump, and you've got Brexit in my country, you've got lots of other issues. Suddenly, everything became tremendously bipolar. You know, you're on one side or the other of every argument. And I can't help thinking this is because social media is a place where moderate opinions don't get repeated and extreme ones do. It's that simple.
And there's and the cat's out of the bag, I guess, or the horse has left the barn on this. There's no changing that. But let me ask about that, because one of the most polarizing figures in a lot of this has been Anthony Fauci. What's your view of him? He's been attacked by the left, by the right. You know, I don't necessarily have an opinion. I think it seems to me he's trying to do the right thing. But how do you view him?
Well, I view him from abroad, which gives me some distance, but it also gives me some caution. You know, I don't have skin in the game of Anthony Fauci's reputation, as it were, in the way that Americans would. I think he's a brilliant scientist. I think he's a very good communicator. I think his distinguished record in particularly the HIV research is something that should be respected. And I think as a public health official, he's been very impressive. But I think on this issue, where his institute did a lot of the funding of relevant research, and he is trying to define what gain of function is in sort of legalistic, semantic terms, rather than simply, come on, did they make a virus that was more infectious or more virulent? Yes, and we know they did in many cases, including in Wuhan. Which is, after all, the only thing that matters. I think on that, it's a pity that he's become so defensive. And, you know, but for me, it's a bit of, I'm a bit worried that America is going to end up having a huge row about whether Anthony Fauci caused the pandemic. Well, he didn't. You know, it didn't start in America. It started in China. Let's not, just because we can't have this, you know, Rand Paul can't question Xi Jinping. Right. So he has to go after Fauci. Do you see what I mean? So, sure. So to some extent, I'm ducking the question by saying, don't let's get distracted. Let's turn this into a US political row, because, you know, we have our Anthony Fauci figures over here in, you know, the equivalent over here in the UK is called Chris Whitty, and a lot of people are furious with him, and a lot of people think he's God. Well, he's neither. He's a fallible human being who's a very good scientist.
Well, let's follow up on, because that was one of the questions I had for you as well. You mentioned gain of function. What is that? What is gain of function? For people who haven't read the book, haven't followed up on this, what is it?
Gain of function became very controversial in around 2014-15, when a series of experiments were done under very careful conditions to see whether you could make an avian flu virus transmissible through the air from mammal to mammal. And the question was, how close are these flu viruses to causing human pandemics? How easy would it be for them to evolve into ones that could infect human beings? And one side of the argument said, we need to do this research to find out. The other side was saying, you're looking for a gas leak with a lighted match here. You're going to cause the very thing that you don't want to do. But the point is, can we in the lab deliberately make a virus deliberately or accidentally make a virus that's more dangerous, that's either more infectious or more harmful. And that's, in the end, what gain of function means. Now, there was a moratorium on funding gain of function experiments,
and they did. They found they and it was they did create a virus. It was much more. It was
absolutely
much more.
In the case of SARS-like viruses in Wuhan, we now know from data which wasn't available at the beginning but has now become available, that in some cases they made viruses that were 10,000 times more infectious and 3 times more lethal to mice by changing their genes. They weren't necessarily trying to do it. What they were trying to do was work out how dangerous is this spike gene if put into this virus.
Right.
And the answer was, in some cases, a lot more dangerous. Now, that's gain of function by any reasonable definition of it. But did it fall foul of the particular moratorium on funding such research, which was only supposed to apply to viruses that might infect humans? These ones, on the whole, were thought not to, etc. You can have an argument about whether it's legally gain of function, which is different from whether it's scientifically gain of function.
But I mean, isn't this one of the key questions? Because there was a piece in the Wall Street Journal, it was December 31st, and it quoted Dr. Kevin Esvelt at MIT, and he was talking about EcoHealth. And I'm going to quote it because, the title of the piece was How This Pandemic Has Left Us Less Prepared for the Next One. And Esvelt said, EcoHealth Alliance should stop characterizing pathogens to determine if they are capable of causing a pandemic in humans. Once they look at each individual virus and characterize it to see if it's pandemic capable, they create a security risk where previously there was not one. But it's following up on your point about this idea about, looking for a gas leak with a lighted match. They're creating a danger by trying to predict which of these, or creating ones to say, well, could this cause a pandemic? Well, it sure looks, there's a very real possibility that that's what, in fact, is at the root of all of this.
Well, I do think that whether this virus came from that happening or not, we need a root and branch reinvestigation of whether or not this is sensible research. Because, going out into the wild, going to bat caves and saying, we've found a bat in here, we've handled it, we've swabbed its anus and its nose, we took our mask off halfway through the process because it was too hot, which quite often happens. We then took the sample back to a city of 11 million people and took it into a lab, and we did experiments on it at biosecurity level 2, which means we were wearing gloves, not much more. This is not a sensible thing to be doing with viruses that you think might be capable of causing a pandemic. Now, when there was an argument in Nature magazine between a bunch of virologists and another bunch of virologists on exactly this topic in 2018. And one side said, this is the way to predict the next pandemic, and it's very important that we do this research. And the other side said, no, this is not the way to prevent the next pandemic. We should be looking at developing better vaccines and things like that. You're not going to predict or prevent the next pandemic by doing this. There's millions of viruses out there. You don't know which one's capable of causing a pandemic, even after you've studied hundreds of them in the lab. And that's what happened. Remember, they studied hundreds, if not thousands, of viruses in laboratories to try and predict the next pandemic. Did they predict this one? No. So at the very least, that research program failed in its own terms. At the worst, it might have actually caused the pandemic.
And that's the part that I thought, as I wrote it down, it's better to pardon the pun, quit monkeying around like this. That this seems like this is incredibly dangerous, and in fact, could explain some of what we're facing now.
Putting it in a very long-term context, because human beings have been going into caves for a very long time. The term caveman, tells you something.
Sure.
And they've therefore been encountering bats for tens of thousands of years. If just the simple act of going into a cave is going to give you a chance of picking up a pandemic, then we'd have harvested every possible pandemic by now. And therefore, the suggestion that in some sense, modern human beings are running greater risks because of encroachment of human society on nature is just nonsense when you think about it. I mean, most of us live in cities now, particularly in China.
Right.
People aren't out in the countryside. They aren't relying on bat guano as fertilizer anymore. They don't need game meat from the forest in the way they did. So actually, the encroachment is getting less, not more, despite the growth in human population, with one exception. And that is, in the last 15 years in particular, there has been a huge amount of encroachment by scientists on wild bat populations in caves to try and study their viruses. That's encroachment.
And that's an interesting way to think about it. Yeah, that the encroachment is going the other way. So yes. Is the virus here to stay? Is this going to be with us forever? I mean, because everyone has been affected by this, whether it's canceled family vacations, Christmas is just ending. Our family plans blew up because of this and, different reasons. But is this going to stick around? Is this just going to be mutating Omicron or Delta, or how long is this going to last? Is there a reason to believe we can see the end of this?
Well,
Please say yes.
I think it's going to be around, but I think we see the end of the chronic phase the acute phase of the pandemic. In other words, we can't go on taking these drastic actions against it. We're going to have to live with it.
And you're speaking about the lockdowns, that we just can't.
I'm thinking about lockdowns. Yeah, I don't think we can go on doing them again and again. And I think it will become milder. Now, that's a relatively unfashionable thing to say. And there was a big pushback when people said Omicron is milder. Well, actually, it turns out it is milder. There are 200 kinds of viruses that cause the common cold. Most of them are rhinoviruses and adenoviruses. Some of them are coronaviruses. Not one of them kills people. I mean, occasionally it can kill a very old person or a very sick person, but basically they are non-lethal. That's no accident. Respiratory viruses spread by sneezes want you is the wrong word, evolve towards ensuring that you are not so sick. They keep their symptoms to relatively mild things that don't stop you socializing, because that way they spread better. And I'm convinced that this thing is going to turn into a common cold. Where did the other 4 coronaviruses that we catch, which cause common colds, come from? Well, one of them jumped into our species around 140 years ago, around 1890, around 130 years ago. And that's when there was a very bad pandemic called the Russian flu. We don't know that it was flu. It might well have been this coronavirus. It started in Central Asia and spread all around the world. It killed 1 million people. It's now a common cold. It's called OC43. You've probably caught it. I've probably caught it in the last few years. It's the commonest of the coronaviruses that cause a common cold. I think that's the fate of COVID. I think COVID is going to become just another common cold. I worry that lockdowns prevented that evolution, because they meant that the mild cases stayed at home while the severe cases went to hospital and caused new waves of infection. That's my worry, is that we've put in place a process that prevents the evolution of the virus.
Would that be forestalling herd immunity then? Would that be the way to think about it?
No, it's forestalling the reduction in the severity of the virus.
Oh, okay. All right. Because it's not going to be immunity. We're not going to be immune to it. We're just going to be less, it's going to be less lethal or less, what's the right word there? I don't know.
Virulent.
Thank you. Well, so let's start, just a couple of more questions. And my guest is Matt Ridley. He's the co-author of the new book, Viral, The Search for the Origin of COVID-19. What is this going to mean, do you think? And you're a man of the world. You've been around for a while. What does this mean long-term for the U.S.-China relationships or China's role in the world? Because it seems, as I've thought about this, and I've written a piece recently about rare earth elements in China, and, COVID is one of the issues of friction, is the South China Sea, the future of Taiwan, the Xinjiang and the Uyghur Muslims. There's a lot in the news about the friction between the U.S. and China. How does this play into this, and what do you see in the future for the how China's role in the world evolves?
Well, I think there's no doubt that my view of China has changed enormously in the last 5 or 6 years, and as most people. The scales began to drop from my eyes when I visited Japan in 2018, and Japanese people said to me, "You don't understand. Xi Jinping is not like Hu Jintao. This is a much more aggressive regime. It's not the free trade thing is not going to work with it forever, etc., etc." So I do think that we are in a much more confrontational phase between China and the West, and that will continue whether Donald Trump's in power or Joe Biden's in power or someone else. I think the economic dependence of the West on China is, I don't see any end to it soon, although I think the Chinese economic engine will sputter. And I say that because I make the case in How Innovation Works that China's economic strength has been freedom, ironically. We don't think of it as a free country. It's not free politically, but economically it has been very free. It's had many fewer of the petty rules and regulations that slow down entrepreneurs in the West. And I think that's ending. I think economic freedom is disappearing under Xi Jinping as well. I think this pandemic will play into that relationship as just another friction alongside Uyghurs and Hong Kong and Taiwan, etc. For me, it's a big one, but for other people, it doesn't seem to be so big. And I'd like to think that it might contribute to a shift from an aggressive to a less aggressive regime in China. But I can't see how that would happen. My parallel here, of course, is with Chernobyl in the Soviet Union. That it did play a part, that debacle, in bringing down a very nasty regime and replacing it with a somewhat chaotic but less cruel regime. And
perhaps less aggressive. Well, I don't know. I was going to say less aggressive, but lately Putin has not.
For a while anyway, that was true. But you're right, no longer so much. It does feel like if definitive evidence of a laboratory leak and the cover-up thereof does eventually emerge into the world, it must, in some sense, weaken Xi Jinping's position. And there may come a point where, in China, people say this man is more of a liability than a benefit. But that's, a lot can go wrong in that. And there are a lot of people who say to me, we shouldn't try and find out the answer because it might lead to friction with China. I'm sorry.
Well, it's an incredible argument. I mean, it doesn't even stand up on any kind of, wait a minute. No, this is the most consequential health crisis of the era. You don't care about how it started? I mean, it seems...
Exactly.
How could you even... It's not a defendable position, in my view. Well, so... do you have another book in the works, Matt? Are you starting on something else? Not that I'm not pressuring you here. I mean, I get that same question.
Yeah, great, Robert. I've just finished a book.
Well, I know you produced it in what, like 9 or 10 months? I mean, it was a remarkably fast turnaround. I mean, I'm not badgering you here, but is this leading to something next?
I think it's much more likely that I will embark with Alina's agreement on a significant rewrite of parts of the book for a paperback edition or something like that.
Gotcha.
But I shouldn't say that. My publishers will crucify me. They'll say, no, please go out and buy the hardback.
It's available at all fine booksellers. The book, again, is Viral, The Search for the Origin of COVID-19. Matt, we've been talking for more than an hour, so I don't want to keep you much longer. And I know you've been working really hard and traveling a lot, promoting the book, and you've just been back from Kenya. What are you reading now? Are you taking a little break? What's on your nightstand? What books do you have at hand?
I'm ashamed to say that while in Kenya, I read books about African birds.
There's no shame in that. I should see the number of bird books that I have in my library here. I collect them all. I think, oh, this is cool. And I go to the used bookstore and I buy some more. And so, reading bird books is very honorable. Don't get me wrong. No qualm about that.
I was given a book as a Christmas present by a friend in Kenya, which he recommended, I think it's called Peacocks and Picathartes, and it's about the unusual birds that live in Africa, and it's really interesting. It's just it's written by an ornithologist, and it's just about weird birds. And I mean, did you know that the ground hornbill has 15 vertebrae in its neck, and other hornbills have 14?
No, I didn't. And
Honestly,
why the extra vertebrae? There just won the sweepstakes to get an extra bone in the neck. What was there a reason for this?
No, nobody knows the significance. It might imply that this is only distantly related to the other hornbills, or something like that.
Well, so I asked you before we started recording. So you said in Kenya you just saw a remarkable number of how many eagles, how many raptors did you see? How many different species? Did you keep a list?
Yeah,
I'm a terrible list keeper. I'm awful at listing.
I'm not normally a twitcher, but
a twitcher. What does that mean?
That's a British term for a bird watcher who's obsessed with lists.
Oh, okay.
But, so just leafing through the fish eagle, the black-shouldered kite, the white-backed vulture, the lappet-faced vulture, the black-chested snake eagle, the martial eagle, huge one,
black-chested snake eagle. Wow.
The battler, the Wahlberg's. No, we didn't see Wahlberg's eagle. The African harrier hawk, the pale chanting goshawk, the auger buzzard, the tawny eagle, the steppe eagle, the Verres eagle, the African hawk eagle, the long-crested eagle, the crowned eagle.
You saw all of these? Holy smokes! I'm going to have to go to Kenya now.
No, you have to go. And there's one on every bush, and they're really tame. You can get within 10 yards of most of them.
It's a very costly episode I'm recording here. That's suddenly. So what you've looked at this issue on the pandemic. And my last question, Matt, as I read the book, I thought, man, this is exasperating. The cover up and the lack of transparency and the horrific toll that it has taken on people all over the world over the last few years. I mean, the loss of life, the loss of income, economic slowdowns, shut-ins, lockdowns. What gives you hope?
Sorry, a dog has just appeared.
Well, a dog is a good thing to give you hope. That's definitely that.
Well, here's one of the things that gives me hope. This pandemic started because of something that went wrong in southern China. Either a market that was selling animals it shouldn't have been selling, wild animals, or a laboratory experiment that shouldn't have happened. Both of those are very easily preventable. And when people say, oh my God, we're living in an age when pandemics are going to become common, they're usually talking about the fact that, there's climate change, deforestation, and things like that. Dogs, will you be quiet? And I'm saying no. Actually, something man-made and exceptional, like the market trade or the laboratory experiments, has got to happen to trigger one of these pandemics. That should give us encouragement, because it suggests that if we don't do those things, we'll be okay.
That if we're more careful, that this is preventable.
Say again?
That if we're more careful, that these kinds of things are preventable.
That's right.
Well, I like that. That's definitely a hopeful note. Matt, thanks for your time. My guest again has been Matt Ridley, Viscount Matt Ridley, the co-author with Alina Chan of Viral, The Search for the Origins of COVID-19. I urge you to go out and buy it. It's quite a remarkable detective story, one unlike any that I've ever seen before. So, Matt, thanks again for being on the Power Hungry podcast.
Robert, thank you for having me. It's really enjoyable talking to you, and great to see you. And come and see us in the UK, please.
I will do that. And to all of you in podcast land, thanks for tuning in. Tune in for the next episode of the Power Hungry podcast. Until then, see ya.